Hi, thanks so much for inviting me to talk. I'm John O'Neill from the MRC Laboratory of Molecular Biology. I'm delighted to tell you a bit about our research, and I'm sorry that I can't be there to present to you in person, but as we all know, there is a global pandemic. So, circadian rhythms, everything you always wanted to know about, jet lag, but were too tired to ask. So, our research to be interesting to anyone that's ever wondered why people tend to feel sleepy around the same time each day, why you get a second wind of energy the next morning if you happen to have stayed up all night. What on earth is happening during shift work to our physiology or jet lag? And so it's all down to this internal biological clock or circadian clock within every cell of our body. The idea that we have internal rhythms in biology is not a new one. And in fact, the first observation was made by Androsthenes of Thasos, an admiral in the navy of Alexander the Great in the 4th century BC, where traveling around the Arabian Peninsula, he noted the daily leaf movements of the tamarind tree, but it required another 2,000 years pretty much until this scientist, Jean-Jacques d'Autus de Morin, also conceived of the idea that there is an internal daily timekeeping mechanism. And this was born of his observations of the daily leaf movements of the mimosa plant. And so he took his mimosa plant and put it into a darkened cupboard under the stairs, perhaps, and every now and then went down and made a note of the angle of inclination of the leaf, and did this over several days and concluded that there is a daily rhythm which is intrinsic to the biology of the plant and not directly driven by daily cycles of day and night. And this is something that's very easy to reproduce with time-lapse photography. So you can see this mimosa plant in constant light in this case, without any idea what time of day it is outside, continues to show roughly 24-hour rhythms in leaf movements, which helps plants in the wild obviously reduce water loss at night. And so the reason that we see these biological rhythms in most organisms on the planet, in fact, is because, of course, we live in a cyclical world due to the 24 hours that it takes for the Earth to rotate around its axis. This is something that affects most aspects of human behavior and physiology. We see it in plants, we see it in experimental organisms. It's something that we can easily study ex vivo. Now, by circadian rhythm, this is dog Latin, and from meaning about daily, and it is used to describe the approximately 24-hour endogenous oscillation in behavior, physiology, metabolism, and activity exhibited in most organisms. You will be most well familiar with the approximately 24-hour rhythm in your own sleep-wake cycle, unless you have very small children, of course. The fundamental features of a circadian rhythm, the thing that allows us to know and to test experimentally, to demonstrate that it's intrinsic to the organism rather than driven directly by daily changes in the world around us, is that it has a persistent free-running rhythm within approximately 24 hours, 24-hour period. So if I were to lock you all in your offices or studies and black out all of the windows and remove all external timing cues, you'd probably be quite irritated with me, but you would continue to show a roughly 24-hour rhythm in when you feel sleepy, when you wake up, and when you feel hungry, when you want to go to the loo, etc. To be of any utility, this biological oscillation needs to be sensitive to external timing cues. And foremost for humans are the times when we see light versus dark and the times when we eat compared with fasting that normally occurs during sleep. And then least well understood is that circadian rhythms are always observed to be temperature compensated, meaning that they don't run faster on a hot day, slower on a cold day. This is not particularly well understood, but these fundamental features of circadian rhythms were first described more than 60 years ago and really remain very much the case to this day. Now, it's something that we observe in pretty much all organisms that you see in the world around you. There are a few extremophiles and archaea where they have not been observed, but certainly the majority of animals, plants, algae, many species of bacteria and fungi express very robust and easily observed circadian rhythms in their activity. And in humans, it's something that we observe at every level of biological scale, from the population level. So here I'm just showing you the adjusted likelihood of having a road traffic accident, which tends to peak in the wee small hours of the morning, because this is when your brain is really not well adapted for sustained concentration and with the slowest reaction times. And so if you are on the road in the middle of the night, adjusting for the number of people that are driving, you're most likely to have a traffic accident. Going down to the level of the individual. So here we're just looking at accelerometry in a single individual over many days, and the data is double plotted. You can see that just like most of us, this individual is most active during the day and tends to be inactive at night. Then down to the level of individual organs throughout the body, which show circadian rhythms in their activity, obviously commensurate with the function of those organs. And so the liver at night is doing night liver things, such as releasing glucose to maintain blood glucose homeostasis, whereas during the day, the liver is storing glucose. But of course, something very different is happening in the kidney and in various parts of the brain. Down to the cellular level. So I could even take a scraping of your skin cells, culture them in a Petri dish, and they would continue to exhibit circadian rhythms when various genes are turned on and off in their metabolism, in their motility, for example. And this would persist for many days ex vivo. And then down to the molecular level, within each of these cells, we know many of the mechanisms and genes which are involved in facilitating these daily rhythms of cellular function. And so in terms of circadian physiology, we often talk about daily rhythms in health overall, because it affects so many aspects of our behavior and physiological function. So for the athletes amongst you, the average individual gives their best performance in the late afternoon, because this is when we see the greatest cardiovascular output and muscle strength. Whereas this surge in cortisol levels in the early morning is the hormonal messenger that really primes us for wakefulness, the thing that facilitates our arousal in the morning, even if the alarm clock doesn't go off. And then the corollary is that there are daily rhythms in the symptoms of many diseases. And so for those that suffer from chronic pain, this tends to be worst in the evening. Whereas if you've ever talked to a doctor that's worked in accident and emergency for a long time, there is a very significant increase in the incidence of heart attacks and strokes in the morning following wakefulness. Whereas if you suffer from depression and you happen to be awake in the middle of the night, this is when the symptoms of depression are felt most severely. But it's not all bad. Of course, because so many aspects of our metabolism, and particularly drug metabolism, are under circadian regulation, this provides the scope for the field of the burgeoning field of chronotherapy, which is the idea that the efficacy of many drugs can be improved by delivering them at the optimal biological time of day. So this was first described for statins, but has now been shown to be the case for quite a few different drugs, including those that are used for hypertension. And the nighttime treatment for hypertension seems to be much more effective than those taken in the morning. Now, the first real observation of circadian rhythms in humans was performed by this brave caveman explorer, Michel Siffre, back in the 60s. And he went and lived in an underground cave system, and by contacting a team on the surface was able to tell them what time he was waking up and how long he'd slept, and obviously monitoring his core body temperature and skin temperature. And you can see that there is a roughly daily rhythm in those parameters. Now, this is not the type of experiment that we really advise people to do anymore. He went a little bit doolally down in this cave system and fell in love with a mouse. And so nowadays we vastly prefer to have individuals in temporal isolation suites where we can make longitudinal measurements of things like core body temperature and cerebral blood flow, which show very robust daily rhythms in the absence of any external timing cues, so that we know that it truly is endogenous to our physiology. Now, you may have heard about the master clock in the hypothalamus, the suprachiasmatic nucleus, and this is the part of the brain which is retinorecipient, and it's the structure which really encodes external photoperiods, so the time when we're expecting dawn, and communicates that to clocks throughout the body, principally via the hypothalamic pituitary adrenal axis, leading to this daily rhythm in plasma glucocorticoids. And so in humans, this signaling network is the thing that leads to the increase in cortisol in the early morning, which primes us for wakefulness and really communicates the timing of anticipated dawn to tissues throughout the body to keep them synchronized with each other and then synchronized with the world around us. Similarly, the SCN communicates with the pineal gland, which secretes the hormone melatonin, which tends to make people feel sleepy. It's one of the things that helps us find sleep more readily in the evening. But sleep is not the same thing as circadian rhythms. The two are closely related to each other, but they can be experimentally separated from one another. It's not a fun experiment, but what you do is persuade an individual to live on a 28-hour day, so 14 hours light, 14 hours dark. And under those conditions, the length of the day is too long for the human circadian clock to cope with. And so it free runs with a 24-hour-ish period, whereas the sleep cycle matches 28 hours, and that allows you to distinguish the two systems. And so current understanding is that there are two processes, process C, which is circadian, mediated by hormones such as cortisol and melatonin, which affects your tendency to sleep, how easily you can go to sleep over the 24-hour cycle. And then another process, let's call it process S, that simply accumulates the longer you've been awake. So this is basically your sleep debt, and that's modulated and mediated by hormones such as adenosine, but is frankly not as well understood as circadian regulation at this time. But in a normal individual, these processes operate in lockstep with each other, meaning that a healthy individual has consolidated sleep at night and is active during the day. You need to be a bit more careful drinking coffee in light of some of the research we've been involved with recently, because of course you often drink coffee to give you a caffeine boost to kind of suppress the symptoms of tiredness, but as well as suppressing our ability to go to sleep by antagonizing adenosine receptors, it also shifts the circadian clock. And so if you drink coffee late at night, you're not only pushing back your homeostatic sleep drive, but you're also delaying your circadian rhythm, meaning that left to your own devices, you will tend to wake up later the next day. And so you're creating an additional problem when you try and awake the next morning. So circadian rhythms really do permeate our physiology. And I'm just going to give you some examples from the liver, because the mouse's liver is probably the most intensively studied tissue in mammals in terms of circadian biology, presumably because even an undergraduate student can dissect the liver from a mouse. So here we find that somewhere between 10 and 20% of all genes that are active in the liver show a daily rhythm in their regulation, as well as about 20% of all liver proteins. And then at least a third of the metabolites in the liver are subject to daily rhythms in abundance, as is almost every aspect of glucose homeostasis. And let's remember that the liver's primary function, or at least one of its major functions, is to maintain blood glucose levels so that we don't go into a coma when we're sleeping at night. Most signaling pathways are under circadian regulation, and even something as fundamental as cell division is under circadian regulation. So if a hepatocyte is going to divide, if it is going to go through S phase or M phase, it will do so at a specific biological time of day. And overall, we know that disruption of internal timekeeping is strongly associated with an increased incidence of many diseased states. And so here we're talking about chronic, usually aging-related diseases such as type 2 diabetes, cardiovascular disease, most neurodegenerative disorders, and various forms of cancer. All of them are associated with circadian disruption, which occurs most frequently in the population, occurs most frequently as a consequence of shift work. And even after a week on shift work, you find a number of individuals that are pushing into metabolic syndrome territory. So going back to this idea that circadian rhythms are observable at every scale, ultimately they are driven by a cell autonomous timing mechanism. They're present in every cell, but their generation occurs at the level of cell biology. And so what do we know about the timing mechanism, these daily molecular timing mechanisms? Well, we certainly know some of the genes. And so this is what the Nobel Prize was awarded for back in 2017. The idea that there are genes encoded in the nucleus which code for clock proteins, particularly the period proteins. These are transcribed during the day and then translated into the clock proteins around midday. Now, these clock proteins first form complexes, and then eventually they are licensed for entry into the nucleus, where they suppress their own expression. They turn themselves off, as well as lots of other clock control genes. And so this transcriptional feedback loop affects the activity of many genes in many different tissues. And this is one of the means by which circadian regulation of physiology is thought to be achieved. Now, what's really cool, and the thing that gets my lab members out of bed, is that this is something that you can really easily observe in vitro. And so here what we're looking at is a fusion of the firefly protein, luciferase, the thing that allows fireflies to glow, fused to a clock protein called period 2. And now these skin cells, these fibroblast skin cells, they're sitting in a microfluidic device with media just gently flowing over them. And so if I start the movie, you'll see that there is a change in light intensity, or bioluminescence, as we call it. And when you quantify the level of bioluminescence over many days, you'll see that these individual cultures of mouse skin cells continue to show a circadian rhythm in clock protein activity. And this is really cool, and gives us a great tool for investigating mechanism, as well as the consequences that this has for our understanding of circadian biology. Because if there's a circadian clock in every single cell of the body, how do they work together? And so current understanding, I'm just going to use a metaphor here of these metronomes balanced on a couple of beer cans. Given the ability to couple with each other by hormonal cues, they are in turn synchronized with the external world by the times when we see light and the times when we eat. And this is the thing that becomes disrupted during jet lag or during shift work, because we are receiving now those feeding and lighting cues incoherently. Now, jet lag, to be honest, I don't have a huge amount of sympathy for, because it's largely a lifestyle choice. If you decide that you want to go on holiday to Australia, you know, there are going to be consequences. But shift work, a lot of people don't have any choice about, and something like, or at least before the pandemic kicked in, something like 14% of the UK adult workforce is engaged in shift work. And so all of those individuals are more susceptible to these chronic aging-related diseases as a consequence of incoherent timing cues, meaning that their individual cellular clocks are basically just doing their own thing. They're no longer communicating with each other. And this is the thing which is thought to put us at increased risk. Now, the mechanism by which the external 24-hour light dark cycle is communicated to cells throughout the body, that's relatively well established. I mentioned it earlier on. But going back just a few years ago, the way in which feeding time was communicated to clocks throughout the body simply wasn't understood. And so a PhD student in the lab, Priya Crosby, she simply asked, how is feeding time communicated to circadian clocks in different cells throughout the body? And she started with the observation that insulin, the hormone that we've all heard of, principally because insulin signaling is deficient in individuals with diabetes, she started off by noticing that cortisol sets the phase of cells to a different time to that which they're set by insulin. So here we're just looking at the same HER2 luciferase rhythm. In black, you've got your untreated cell control. And then if you give a bolus of insulin or cortisol, you can see that the two cellular populations are at different circadian times. That means that insulin is having a different effect on circadian rhythms than does cortisol, the hormone that we already know about, which synchronizes them with the timing of dawn. And Priya went on to show that under these perfusion conditions, you just provide a short bolus of insulin that simulates the increase in the levels of this hormone in the blood after you've had a meal. That was sufficient to completely reset circadian rhythms in these cells, comparing the red traces with the black trace. And then with our colleague David Bechtold in Manchester, he showed that the same was true in a mouse model. So we get this acute induction of the HER2 clock protein and then a delay in circadian rhythms the following day simply by injecting the mouse with a physiological dose of insulin. And so I'll skip a lot of the data, but the model that we arrived at is that when you eat your first meal of the day, the increase in insulin signaling leads to a signal that propagates intracellularly to increase the production of the period 2 protein. And this is sufficient to reset or to set the timing of individual cellular clocks to coincide with the timing of that first meal throughout the body. So that's an example of some of our research. What else do we do? Well, because circadian rhythms are intrinsic to mammalian cell biology, we start a lot of our investigations using cultured cells and then work up to understanding the physiology or work down to understand the molecular mechanisms. And so our primary question, I guess, is what is the cellular timekeeping mechanism? How on earth does an individual mammalian cell or human cell time 24 hours? But of course, to be able to answer that, we need to understand all of the external timing cues that allow these cells throughout the body to be synchronized with each other and set to resonate with environmental timing cues. And then, of course, the really translationally relevant question is, how is circadian regulation of biological function achieved? And clearly this varies enormously depending on the cell type that we're studying. In terms of the cellular timekeeping mechanism, I will not bore you with the details. Suffice it to say that this is a work in progress. Many of the relevant genetic mechanisms have been identified, but the biochemical mechanisms, the enzymes that act upon those clock proteins to drive 24-hour rhythms in their activity, are still not really understood. And we know that you don't need these cycling clock genes in order to sustain a circadian rhythm. Because once upon a time, we looked at human red blood cells isolated outside of the body. And astonishingly, they continue to exhibit circadian rhythms in biological processes such as ion transport and protein turnover that we see in other cell types as well as in vivo. And in all cases, we find that the speed at which the cellular clock runs is determined by the activity of enzymes such as casein kinase 1. And so maybe if you invite me back in another 4 or 5 years, I might have a more complete mechanism to tell you, but this is something that we are very much actively engaged in trying to understand. And then in terms of how circadian regulation of function is achieved, one example is a great former postdoc in the lab, Ned Hoyle. He found circadian rhythms in the cell's cytoskeleton, in the actin and actin regulatory proteins. And so this is the structure in the cell that allows them to move, that allows skin cells to move, to migrate and to adhere to each other. And so because of this rhythm in the cytoskeleton, Ned tested the hypothesis that this should have an effect upon wound healing, because of course, if you sustain an injury to your skin, skin cells need to be able to migrate into that gap, and also to be able to remodel the blood clot. And so this is what he found in cultured cells, that wounds incurred during the active phase, the cells migrate much more rapidly than those incurred during the rest phase. And then in actual tissue from mice, you can see that 48 hours after a wound was incurred in this three-dimensional confocal reconstruction, the fibroblasts in red here, the primary protagonists of wound healing, they have migrated far further into the wound, and there are far more of them in the wound than for those wounds that were incurred during the rest phase, when the mouse would normally be asleep. And so based on our cellular and mouse data, we arrived at this model that led to the prediction that the same would be true in humans. And so we were very fortunate to be able to be given access to an NHS database of burn injuries sustained to patients. And we simply tested our prediction that the same which we observed in mice and in cells would be the case in humans. And this is what we found, that burn injuries sustained during the active phase, during our daytime, heal significantly faster than those which are incurred at night. More recently, we've been looking at how cells deal with daily changes in protein concentration. Now, the cytoplasm, the interior of a cell, is really jam-packed with molecules like proteins and smaller ions such as potassium and sodium and chloride, as well as lots and lots of water. Now, cells really don't want to change their volume, but there's only so much room inside the cytoplasm. And I told you earlier that the amount of many cellular proteins is controlled by their cellular clock. Now, it turns out that the overall concentration of protein in the cytoplasm changes by about 20%, and cells compensate for this by exporting ions through transporters in the plasma membrane. If you imagine that the cell is like a dance floor at a wedding, right? The ions are the children and the proteins are the grown-ups. Initially, there's a lot of children and very few adults filling the dance floor, but as time progresses and the DJ puts on some better music, more adults feel the need to boogie, and so the children leave the dance floor to accommodate them. And so this dance of proteins increasing, ions leaving the cell, proteins decreasing, ions coming back into the cell is happening in all of our cells every day. Now, the concentration of sodium and potassium is important for heart cell function, and this affects how fast they can beat. We find that this ion rhythm that we see in all cell types is actually large enough in isolated heart cells that it confers a daily rhythm upon their electrical activity. So when ions are high at the beginning of the day, when the mouse is just about to be most active, we see that the capacity, the cell intrinsic capacity is to beat is much higher than at the end of the day when the heart would normally be resting. And this is something that we see in isolated heart cells, as well as in mice in vivo. And for the electrophysiologists among you, of course, heart rate is regulated by the autonomic nervous system, but we have found that this heart intrinsic rhythm in firing activity persists under autonomic blockade. So basically, compensatory ion transport facilitates daily changes in cellular protein content. And in the heart, these rhythms allow cells to beat faster at the biological time when they need to do the most work. So these are the questions that we're trying to answer. What's the cellular timekeeping mechanism? How are extracellular timing cues communicated to clocks throughout the body? And how is circadian regulation of biological function achieved? Only when we understand how this cellular clockwork interacts with all the other systems of the cell can we really start to understand how daily changes in physiology are achieved, and thereby prevent or even reverse the temporal dysregulation that occurs during shift work and aging to promote health and the improved treatment of disease. And so the take-home message is that every cell and tissue in your body has a circadian clock, and this confers daily rhythms upon the activity of those cells. Normally, these rhythms are all in synchrony with each other and organize our physiology to anticipate the differential demands of day versus night. But when circadian rhythms get out of sync with each other, there is an increased risk to health. And so to avoid this, paying attention to when we eat and the times when we see light is a really good idea, as well as avoiding bright light at night and fasting, in order to keep those circadian rhythms throughout the body synchronized with each other and with the external cycle of day and night. And this is a relatively recent photo of my wonderful lab members, and I'm really looking forward to hearing any questions that you might have.